Thank you so much for warm introduction. So it is a really great pleasure to be here again and then present our latest data focusing on pathways balancing mucin and CFTR-mediated fluid secretion, especially focusing on human distal airways. As Julie introduced, unfortunately, my flight was delayed, and then although I left like 6 a.m. Eastern time in North Carolina, but I just landed 30 minutes ago and then changed clothes in Uber and then just finally made it. And I'm so reassured. So, yeah. Thank you. So I have no conflict of interest. So starting with my presentation, so let me introduce the unique structure of the lung. So the lung, especially airway, has a unique structure, and then airway is branching and branching and branching and 23 times to connect to the alveolar region. And then if we can classify the large and small airways, the classical definition of diameter of airway is 2 millimeter. So, and then there are different structure between large and small. One is cartilage and another one is hemicycle glands. So these structures are only present in the large airways, which is more than 2 millimeter of diameter. And then small airway doesn't have those structures. And then, yes. However, interestingly, because surface area and the number of small airways exponentially increase in distal airways because it's branching and branching. So if we calculate the proportion of total surface area from large airway and small airways, actually most of the surface area in the airway is occupied by small airways, like 98%, and then 1.6% dominated by large airways, which is also called like bronchi. And the small airways are usually termed bronchioles. And this is interesting because if we have some bad thing happens in large airways, which consists of only 1.6%, unless you are choked, usually it's fine, like upper respiratory virus infection. But if you have something really bad in the small airways, so then most of your small airway surface area is affected. So that makes the lung function really severely damaged. And then indeed, CF does affect small airways. So this micro CT image data show mucus obstruction starts 6th generation bronchi to all the distal airways, and that proportion is increased in the distal airways. This like gray part is a mucus obstructed airway number, and the white part is a still open airways. So this micro CT image shows like mucus plug in the very tip of distal airway, which is called terminal air bronchioles. Also, our group's data shows like secretory mucin component is also altered in the CF small airways. So usually, this very tip of like distal terminal bronchioles are almost mucin free, but in CF, this green MAC5B and also MAC5AC expression is increased in the CF small airways. That leads mucus plugs. Also, lung function data shows, interestingly, that if you compare normal subjects and the CF subjects for lung clear function, in central lung clearance, there is no significant difference, but in the peripheral lung clearance, which indicates small airway, includes small airway clearance, is significantly reduced in the CF. So these data suggest CF does affect small airway region. Also, obviously, other bunch of data suggest CF affect not only small airway, but also large airway surface area and the hemicycle glands. So all those components are affected by CF. But today, so I'd like to just focus on CF, small airway regions. And then also, not only CF, but also other mucobstructive disease is actually affects small airway as well. So this is asthma and COPD. So the CT scanning shows mucus obstruction in the small airway regions, and that correlated with lung function decline in the COPD. And also in asthma, so a little different from CF, but the MAC5AC, which is a more stickier mucus, like secreted mucin, is significantly increased in the asthma small airways. So these data suggests like small airway region may be vulnerable to mucus obstruction. So the part of those reasons may be the lack of submucosal glands, so which is very important to flush out the mucus in the large airway region. But in the small airway region, as I mentioned earlier, so there is no submucosal gland. So that's a lack of supporting system. And also airflow, for example, like cough clearance is efficient only in the large airways. So those things are probably contributing to small air vulnerability to mucus obstruction. But there are not so many papers reporting the region-specific mucociliary clearance in the lung. But I found some evidence. So the mucociliary clearance is slower in human small airways compared to large airways. And another evidence is mucus is less concentrated and less viscoelastic in the canine small airways compared to canine large airways. And also cilia, the ciliated cell frequency and the length of cilia is less and shorter in human small airways compared to large airways. So these are at least like the evidence which has been reported. And then if these are all the case, so why is mucus in small airways diluted and slowly transported? And then we think about the benefit of this because the small airway surface area is exponentially big compared to large airways. So if this system might be helpful to avoid accumulation of mucus and obstruction in more proximal airways to accommodate the exponential actually decrease from distal to proximal in airway surface area. But also we hypothesize that this system might be important for the mucus transport from all big surface area, small to large airways, but also this might have a cost. The dilute mucus layer and the slower clearance rate may lead to vulnerability to inhaled toxicants in small airway regions because like a longer exposure to inhaled toxicants could happen in this system. So this potentially different mucociliary clearance regulatory mechanism may be also another reason of like small airway vulnerability to mucus plug. So that's our hypothesis. And another interesting thing, and that which is very relevant to CF pathogenesis, is a CFTR regional distribution. So our previous study shows CFTR expression is relatively constant from large to small airways. Another interesting finding along with this data is like ionocytes. So ionocyte is a recently identified rare airway epithelial cell type, but which has the highest level of CFTR expression in airway epithelial cells. And this data shows like, while CFTR expression is relatively constant, FOXI1 positive ionocyte is enriched in the submucosal gland duct and also large airways, but it's becoming sparse in the distal airways. So there is a gap between frequency of ionocyte and then CFTR expression, especially in the distal airways. This data is actually reproduced by other group in newborn pig. So in newborn pig, the same finding was observed, like only in large airway surface, probably submucosal gland duct, ionocyte was identified, but virtually no ionocytes in the small airways. So these data might suggest like other than ionocyte is likely to be contributing to CFTR expression and function. Therefore, mucociliary clearance regulation in the small airway region. Related to that data, so more recently last year, two groups report a paper focusing on small airway unique secretory cell populations. And then now they have more finely analyzed small airway epithelial cells, and then identified smaller unique population expressing SGB3A2 and SFTBB. So as shown in this histology pictures, so you can see SFTBB and SGB3A2 expression only in bronchiole, which foot diameter is less than 2 millimeters, but not expressed in the larger airways. So, and then more importantly, this population also is a predominant cell type expressing CFTR in small airway region. So this is consistent with our data showing a gap between CFTR expression and then ion site frequency in the distal airways. And also this is consistent with our single cell RNA sequence data, which was also published. So then now we know in small airways, probably single cell data suggests different airway epithelial population reside in the small airways compared to large airways. And also mucociliary clearance may be different between large and small. And the mucociliary clearance is in general regulated by airway epithelial cells, and that population is different. So in that case, it's very important to understand small airway specific mucociliary clearance regulatory mechanism. And then we need to study that function, because small airway region is very difficult to access. And then animal model, in both animal model and in human in vivo. So in vitro model is a good tool to measure small airway epithelial cell biology and physiology. But conventional HB cell cultures usually originate from large airway stem cells, like basal cells. But this is great too. So this is nicely recapitulate in vivo pseudostratified airway epithelium. However, if we go back to this estimation, so this large airway may represent 1.6% of total surface area in the airways. And then 98% of surface area is dominated by small airways. But this, and also now we know there is at least a difference in the epithelial cell population between large and small. So I think many things may be common between large and small airways because they are still both airway epithelial cells, but also it's not known if all the finding is really applied to small, all the findings identified in the large airway culture can apply to small airway. So then to overcome that limitation, we have developed a large and small airway in vitro culture system. We isolate large and small airway stem cells from different regions. And also we use 2 different techniques to isolate small airway basal cell populations, stem cell populations. One is microdissection, and another way is we just cut the peripheral lung piece and then chemically digest everything and then seal them and then select using the media. Then after conference, so we transfer them to air-like condition and then let them differentiate it for 1 month. Then this large and small air culture shows a little different phenotype. This large and small, this SFTBB, and SFTBB expression is highly exclusive in the small air in vivo, and then this culture system also recapitulate that difference. And also the good thing about this bulk digestion method is that we can isolate small airway stem cell population from any disease lung. It's really hard to microdissect CF small airways because of the inflammation and then attachment to the surrounding parenchyma. But this method, we can cut a very peripheral lung piece of lung and then exclude any large airways, which is virtually more than 2 millimeters of diameter. So then this data shows nice similarity between microdissected small airway and the bulk digested small airway culture. And also we were able to culture CF small airways as well. And also we perform bulk sequence, and also like we tested 2 different culture media, UNC ALI culture media and the pneumocult culture media. And then there is a little bit different gene signatures, but still largely small airway specific gene signatures preserved regardless of media, and then different from large airway specific signatures. So these data suggest like our small airway system is retaining the in vivo feature of small airways and then distinguishable from large airways. So then now we can use this system to measure mucociliary clearance parameters. And firstly, we looked at % solids in the mucus, apical mucus from these large and small cell cultures. And the large airway apical mucus is more concentrated, shows higher % solids compared to small airways. And then this is consistent with the Kenichi small airways data. And then when we look at solid component and the fluid component, actually solid component is comparable, but fluid component is higher in the small airway culture compared to large airway. And then I thought initially like this might be leaky because small airway culture is a little thinner than large airways, but actually resistance is comparable. And also like mucus viscosity is lower in the small airway cell culture. That's correlated with % solids data, and then consistent with Kenichi data. So, and then these like rheological data suggest small airway cultures are more diluted by more fluid secretion. Then next we looked at CFTR function between large and small cell cultures, and then we perform like around more than 10 calls, and then co-duplicate per each call. And then small cell culture shows about 2-fold higher CFTR function. And then this is correlated with CFTR mRNA expression. So this data suggests probably that like more dilution observed in small apical mucus may be associated with higher CFTR function in the small airway cultures. Then the next question is how that difference in the CFTR function happens. And then we performed single cell RNA sequence to make sure which cell type express CFTR in our cultures. And then this data shows contribution to total CFTR expression. So this is defined by the expression levels and then frequency. So we calculated the contribution of each cell type to total CFTR expression. The secretory cell population consists of about 50% of CFTR expression in large area, and then that proportion is increased to 66% in the small area. And the rare cell population include ionocytes, and then that contribution is also decreased to the small area cultures, which is consistent with the in vivo data. And this is a violin plot showing this is secretory cell population express the most of CFTR among all the epithelial cell populations. And this is also, this SGB32 is supposed to be only expressed in small cell culture. And then there is a 2 column here, and the right side is small airways. So as we expected, this SGB32 is only expressed in the small cell culture, and especially in the secretory cells. So that also reassuring. So our small airway system is similar to in vivo small airway epithelial cells. And then further to confirm this secretory cell population express CFTR. So we have generated a lentivirus system carrying rat CCSP promoter driving GFP to label only CCSP positive secretory cells in the primary large and small cell cultures, and then perform the FACS sorting to select only GFP positive cells. And firstly, this hormone immunochemistry shows nice specificity of our rat CCSP promoter to the CCSP positive non-ciliated cells. So this is a dome-shaped cell sustained for CCSP and GFP. And then we looked at the mRNA expression of marker genes. Secretory cell marker genes are highly expressed in the GFP positive cells in both large and small cultures, so as we expected. So because CCSP is used to be thought the club cell marker, and the club cell is a predominantly exists in the small areas, but now CCSP is actually expressed in both large and small area cell cultures as small area secretory cells. So, and then, but CFTR, importantly, CFTR track this secretory cell marker expression. CFTR is expressed more highly in the GFP positive cell in both large and small. And the basal cell and the ion site markers are actually opposite, highly expressed in the GFP negative cells. So that means probably CFTR expression is more affected by secretory cell populations. And further, to make sure if secretory cell can be is capable of expressing functional CFTR, and then we replace the GFP with wild type CFTR, and then infect CF large and small cell culture with this lentivirus to selectively restore CFTR, wild type CFTR protein in the secretory cells. Then again, the immunohistochemistry shows the co-localization of CFTR in the CCSP positive cells, and then these are non-ciliated cells. And then this RAS CCSP promoter driving CFTR transduced cultures show wild type mature CFTR protein. And then using chamber data shows nice restoration of CFTR function in only CCSP promoter driving CFTR infected cultures. And then this is a like visual image photo of cultures. So you can see like fluid secretion in the CCSP promoter driving CFTR infected cultures. And another interesting thing, so we identified in the single cell and sequence data is because now secretory cell population, especially in small airway, is important for CFTR expression. And if we look at the pathway, which pathways enriched in this population. And then one of the enriched pathways, the IL-1 pathway. And then there is a couple of reports showing IL-1 beta stimulation increase CFTR expression and CFTR function in cultures. And also another data from UNC group shows if we treat HB cells with supernatant of CF sputum, so then the modulator efficiency is actually up in those like little pro-inflammatory condition. So those data led us to think about IL-1 pathway may be already activated in the basal condition, and then that could regulate CFTR function. And also it's known like IL-1 beta is a pro-inflammatory cytokine that regulates mucin secretion. So, and then now we know secretory cell produce mucin, and now we know express CFTR. So then IL-1 pathway may regulate both mucin and CFTR expression in a single cell type. So, and also another supportive data is IL-1 receptor is highly expressed in the small cell cultures compared to large airways, and also predominantly in the secretory cell. So this might suggest like the IL-1 pathway is already activated in the basal condition, especially in the small area cells. And then we have looked at the IL-1 receptor expression in the in vivo tissue sections. And then we can see a nice causalization of IL-1 receptor 1 with CCSP and SFTBB, positive small airway secretory cell population. And this is a site spin plus ion RNA scope data showing CFTR expression with IL-1 receptor expression, and also these cells are labeled as SGB382, which is a small secretory cell marker. Then test this hypothesis. We add IL-1 receptor antagonist to the small cell culture, and then look at the MAC5B and also like IL-1 pathway downstream gene expression. And this gene expression is reduced by IL-1 receptor antagonist even at baseline. So this suggests already pre-activate IL-1 pathway in the small cell cultures. And then the more importantly, when we add IL-1 receptor antagonist in the smaller cell cultures, so the CFTR function is reduced to the almost half. And then in contrast, when we add IL-1 beta to the cultures, so the CFTR function is increased. But this increase is also offset by IL-1 receptor antagonist here. So this suggests IL-1 pathway is likely at least partially regulate baseline CFTR function. But also if we block this IL-1 receptor, but still CFTR functions remain. So maybe another pathway, other pathways are also contributing to baseline CFTR function. Okay, so then the last topic is now we are focusing on small airway secretory cell as a very important cell type in the small airway mucociliary clearance regulation. And then if we go back to CF lung tissue sections, and then it's already known, MUC5AC and MUC5B, these are secretory mucin. Genes are upregulated in the CF small airways, but also this spatial transcriptomics data shows STB310 and SFTPB, these are small airway secretory cell markers are also in contrast reduced in the CF small airways. So this suggests in CF small airways, the normal small airway identity might be lost in the CF small airways, and that might be associated with CF small airway pathogenesis. Then we pick up like one transcription factor which might be important for regulating those small airway cell secretory cell identity, because this NKX2.1 is known to be very important in transcription factor during the lung development. And also even in adult, this NKX2.1 expression shows a gradient from large to small airways. And then in small airways, this transcription factor is high in the small airway epithelium cell and also AT2 cells. So we focus on this transcription factor and then perform the CRISPR knockout of this gene in both large and small airway, primary airway epithelial cell cultures. And this is a control cultures showing nice seriated cells in both a little less, but small airway cultures. And then these white positive cells are SFTPB positive cells. And then this is only found in the small airway cell cultures. And when we knock out NKX2.1, so almost no change in the large airway cultures. But in the small airway cell cultures, there is massive increase in the MUC5AC positive goblet cells. So, and this is a histology of small airway cell culture, NKX2.1-KO. And then we quantify the number of area positive for MUC5AC or SFTPB. So MUC5AC positive cells are significantly increased only in the small airway cell cultures. And then SFTPB positive normal small airway secretory cells are significantly reduced in the knockout cells. And also to further confirm these findings, we have performed Balkan sequence to look at the goblet cell genes more globally. And then goblet cell genes are highly upregulated in the NKX2.1-KO small airway cell cultures, but no change in the large airway cultures. And then this is a small airway secretory cell genes only expressed in small airway cells in control, and then no expression in the large airways, but these genes are globally downregulated in the NKX2.1-KO. So this suggests that NKX2.1 is required to maintain this very important small airway secretory cell population. And then abnormal dysfunction of NKX2.1 induced goblet cell metaplasia in the small airway cells. And then interestingly, CFTR expression is also decreased when we knock out NKX2.9-KO in smaller cell culture. That suggests CFTR is likely to favor normal condition rather than goblet cell metaplasia conditions. And then when we look at the other cell type markers, other cell types are almost like not affected by this NKX2.1-KO, like ion site seriated basal cell population. So this NKX2.1 really pinpointedly regulates secretory cell in the smaller cell cultures. So this this is my last slide, but this data suggests if we think about restoring CFTR function in CF small airways, it may be important to restore normal homeostatic small airway epithelial cell population first. Then if we restore CFTR gene molecular therapy or modulator therapy, so then it might be more effective to restore CFTR function. The summary, mucociliary clearance mechanism is likely different in small airways compared to large airways in human. And small airway secretory cells are key cell type regulating small airway mucociliary clearance. The IL-1 pathways at least partially involved in maintenance of small airway mucus homeostasis. And NK2.1 regulates specificity of small airway secretory cells. And the human in vitro small airway culture is a very useful model to study small airway specific epithelial biology, physiology, and then muco-obstructive lung disease that affects small airways. Yeah, I'd like to thank all my lab members and also my previous mentors, Dr. Boucher, Wanda O'Neill, and Scott Randall, and also outside collaborators. And then, of course, I'd like to thank all CFRI and other foundation which has been supporting my project. I remember, since I just joined Dr. Boucher in 2016 as a postdoc, and then the first grant, fortunately, I have funded is a CFRI grant, and then since then, so CFRI has been really supporting my research, and then this year finally I have got like my own R1, so yeah, I really appreciate CFRI as well. Yeah, thanks so much. I'd be happy to take any questions.